Hey, welcome to Modern Day Debate. Very excited to have you here. It's been a wild week, and this is about to get wilder. So we have got two gentlemen here, experienced debaters, and they are going to basically be giving their broad case for their world. We could say maybe scientific explanation of origins would be the best. They would both say that science backs their view, and so we'll let you be the judge, crowd. We're excited to get your questions during the Q&A, and we're excited to have the debaters present their cases. So without any further ado, we are going to just quick give a brief synopsis of what the format's going to be for tonight. We're going to have 15-minute introductions from each, basically an opening statement from each debater, followed by a 10-minute rebuttal from each debater, followed by a 5-minute closing from each debater, and then the Q&A. So I'm going to let each debater just share a little bit about what they've been up to at their channel. And then after that, we'll jump right into the debate with the opening statements. So for that, we would like Jackson to first share what he's been doing at his channel. Jackson Wheat, we're really glad you're here. Thanks for coming back on, as you've been here before. And the floor is yours to share about what you've been up to at your channel, Jackson.
You have the power to unmute my microphone. Oh, man. So powerful. I have really not been doing a whole lot, I'm sorry to say. I did a talk with a flurfer the other day with RJ Downard the other day. That was like, was that before Christmas? Who cares? Anyways, I did a talk with a flurfer with RJ. I was doing a series of videos on the phylogenetics of dinosaurs, of course, leading up to aves, the modern dinosaurs. And that's pretty much where it is. I've just been lazing about recently, and so I haven't really been doing any YouTube stuff. And also preparing for this. That's taken the most of my time. So there you go.
Excellent. Well, and we're glad for that. I'm always impressed by the amount of preparation the debaters take this seriously. And so I know that you guys have taken this seriously. You've hit the books and you're ready to share your stuff. And that's why I just appreciate the debaters so much. And next, we have Standing for Truth, who's just going to share a little bit about his channel, what he's been up to. Standing for Truth, thanks for coming back on. We're glad you're here.
Thanks a lot, James. Thanks for having me on. Jackson, thanks for participating in this debate with myself. I'm kind of like Jackson, nothing too crazy going on my channel. I think a few discussions, if anyone's interested in those, mainly the creation evolution topic. Mostly just been enjoying your channel here, James. You've put on some really good discussions, really good debates lately, so I want to commend you for that. You're running a great channel. I recommend them to everybody, so I'm looking forward to more of these. But other than that, you know, I've been spending most of the time like Jackson preparing for this debate. Just want to give people a good time, a good show. So I'm certainly really excited for it. I think it's going to be fun. Thanks, James.
Thank you. I appreciate your kind words, and thank you for everybody's encouragement recently. People have been encouraging and helpful. It's so helpful. I love when you guys give feedback. Sometimes I get an email and it's like, "Oh, hey, you could probably do this with your audio. It'll be better." Or, "Hey, you could use Cirrus. Let me know. Hey, you could actually use an Ethernet cable. That'll give you a boost in your internet." Like that kind of stuff is so helpful. This channel like truly is like an abstract entity. Like it doesn't belong to me. It really belongs to all the people that have been so helpful. I'm so thankful. And this debate is appropriate following the last debate we had with Kent Hovind and Inspiring Philosophy. If you haven't seen it, encourage you to check it out. And also, when we had those technical difficulties, I am so thankful, both for the debaters were so kind, they were so gracious, but also that people on the web, like on Facebook, were like sharing and they're like, "Oh, hey, here's the new link." And they were helping out because I was like, "Oh my gosh, this is a mess. What's going on?" And so I just appreciate so much, so many people helping during that tough time, sharing the links and stuff like that. And if you enjoy debates, hit that subscribe button. We've got plenty more to come, and we're working on advances in the channel, like the software, stuff like that. We're still working a few bugs out of, but we're stoked about the future with a lot of awesome stuff, hopefully with these debaters returning. Last thing I'll say before we get to the actual opening statements, both of the debaters have their stuff both of the debaters have their links in the description box below. So I would highly encourage you to check that out. Even if you don't agree, I would say that you should still check out their channel, because then you can at least understand where they're coming from. So with that, I am going to start the clock. As mentioned, this is a formal debate. So we are going to start it with a 15-minute opening statement, then followed by a rebuttal of 10 and a closing for 5. And finally, we're going to wrap up with our Q&A. Any leftover time that we have during the statements is actually going to be added to the Q&A. So we're excited for your questions. Shoot them in the comments if you have them with an @ModernDayDebate, just to let me know that they're meant for one of the debaters. Standing for Truth is going to lead us off. We're very happy to get this ball rolling as I reset the clock. Standing for Truth, when you are ready, the floor is yours.
Awesome. Thanks so much, James. Just want to make sure that you can see the screen. Is the screen being shared okay there, James?
It is definitely being shared perfectly well. Thank you.
Awesome. Okay, I think I am good to go. Thanks so much again. The debate is biblical creation or evolutionary theory. What does the data reveal? Now, is evolutionary theory so well established that it's compulsory to adopt evolution as the means for the origin of species? Let's start with some definitions. I think that's important. The term evolution can mean very different things. I think Jackson and I can agree on that, such as change over time, or in regards to biological evolution, a change in allele frequency in populations over time. Change over time and change in allele frequencies are not really disputed. The type of microevolutionary variation where you get a change in the frequency of the expression of different traits, this type of evolution explains the survival of the fittest, but not the arrival of the fittest. It explains the small scale variation, but not the major innovations, the major origin events of major new forms of life. Change over time is pretty much uncontested. Now, universal common descent, on the other hand, is an entirely different matter. The best way to represent it is the way Darwin himself so did in The Origin of Species. It's a great branching tree where the branches at the top of the tree represent the major life forms we see all around us today, while the side branches may represent various forms of life that went extinct a while back. And the root, very far back in biological history, from which all the various biological forms of life are thought to have evolved or morphed or changed from that one simple form. Essentially, this is a theory of biological history, and that there has been very gradual and continuous change in life leading to and producing the various forms of life we see today from that one or maybe few simple ancestral forms. The big problem, though, and the major issue here today is the cause of this type of change, whether be a change over time or a change in allele frequency in populations over generations. Now, is there a limit to the change? I think that question's quite obvious. Evolutionists would say the cause is an unguided process of natural selection acting upon random variation or random mutation. I think it's absolutely clear and evident that these processes of natural selection and mutations lack significant creative powers. The idea that an undirected, unguided process can produce all the various forms of life in all their incredible complexity, giving the illusion of design, is absurd and non-science on very many levels. So if by evolution you mean dogs and wolves are related through common ancestry, then that's not the issue, because I agree. But when you say that by evolution you mean dogs and banana plants or dogs and whales are related through common ancestry, and that there's actually evidence for that, I say you must be strongly anti-science and anti-common sense, because all new innovations, structures, body plans, and novel information cannot be the result of natural selection acting upon random mutation, random variation. Why? Because neo-Darwinism lacks incredible explanatory power in regards to the origin of phenotypic complexity, anatomical novelty, and the origin of non-gradual modes of transition, aka abrupt fossil appearance. And so what is the real origin of species? And why does the biblical model better explain the empirical data? We know the question of the origin of species concerns the origin of traits. not bones found in the dirt. Since we know traits are inherited, Mendel was the first to expound the principles of inheritance. Darwin knew nothing about genetics. The origin of species and traits is a fundamentally genetics question. Now, do you think God intended the command, be fruitful and multiply, to be by cloning? If you think and say to yourself, wow, that is weird, what a strange question. No, of course not. Then you've basically acknowledged a particular and very specific genetic view. This view, if you're looking at the slide here, is that Adam and Eve were created with pre-existing DNA differences. This is the created heterozygosity hypothesis. It is both testable and falsifiable. Mutations consist of very little of the DNA differences, and inherited DNA differences contribute a lot. This hypothesis is both consistent with mitochondrial DNA rates and nuclear DNA rates, as evolutionists have over-predicted one and under-predicted the other. Both DNA compartments are reconciled perfectly under a creation model. The signature of this hypothesis would be an enormous amount of DNA differences. Now, if you can picture God's original created kind somewhere between the rank of subgenus and order, these kinds contain millions of heterozygous sites in their genomes. As they reproduce and fill the earth, shifts from heterozygosity to homozygosity led to diverse offspring. Less than 1,700 years after creation, there was a flood bottleneck where populations were reduced. But because this population bottleneck was so short, the heterozygosity of the Ark passengers would have been minimally affected. For sexually reproducing kinds, a male and a female could have possessed a combined 4 copies of nuclear DNA. These copies would have been very different based on pre-existing and functional DNA differences, preserving a massive amount of speciation potential. In other words, with incredible design and functional heterozygosity, even at only a tiny
Standing, we lost you. Standing. Standing, we lost you. We can't hear you.
Limited time diversity. Oh, hear me now?
Yes. If you want to maybe like just jump back like 15 seconds into your speech, we can give you that, because it clearly was a connection problem.
Like right when the words came up, we lost sound.
You mean the whole time?
No, only
just for the past few, like 10, 15 seconds.
Oh, okay. Are you able to
yes, we can hear you now.
Bring back the clock 15 seconds?
Yeah.
Okay, perfect.
15 seconds. And ready. Go ahead right now. Thank you.
In other words, with incredible design and functional heterozygosity, even at only a tiny fraction of their nucleotides, through the processes of recombination and gene conversion, an essentially unlimited range of useful diversity would be produced. Now, I've heard evolutionists say, does he not know about linkage groups? This statement exhibits a strong misunderstanding of our model. For one, these designed variants would be expected to be created within useful linkage groups and would have originated at high allelic frequencies. And so it is all the more obvious and consistent that with heterozygous ancestors, an almost limitless variety of combinations of chromosomes, genes, and traits are possible. Linkage groups, just a quick detour here. The human genome is made up of linkage groups, and a typical linkage group is 30,000 letters long. Mutations in that linkage group are linked basically forever. And so if you have lots of bad mutations in that linkage group, and that linkage group is reducing in fitness continuously based on accumulating mutations, deleterious mutations, and you throw in 1 or 2 beneficials, they are just going to get neutralized by the large number of deleterious ones. The linkage is actually a killer for evolution. This means that every linkage block is degenerate. Linkage groups can only get worse and really not better. This means evolution cannot be true and is therefore false. Once again, genetics proves biblical creation. Now, more on genetic degeneration later. I want to get into the LMN haplogroups, because it is widely known that human mitochondrial DNA differences divide into 3 major groups, designated L, M, and N. These are the 3 major haplogroups. The history of humanity is in our genome. What we know about mitochondrial DNA of humans fits the biblical-based model. The model explains the 3 haplogroups and the relative differences between pre and post-flood humanity. Mitochondrial DNA is inherited from mothers. There were 3 young women on the ark who would have repopulated the earth after the flood 4,500 years ago. The fact that there are 3 major haplogroups of human mitochondrial DNA in the world is amazing confirmation of this model. Also very consistent is the Babel dispersion, where one would expect sudden formation of several variants in human mitochondrial DNA due to rapid separation and isolation of various groups of people. On this tree and map of humanity, the 3 major haplogroups are joined by relatively short line segments, indicating that there is not much difference between them. This is consistent with the pre-flood world of only 10 generations, long ages, being that there are little differences between them. Now, these women came from a world where only a few generations had lived before them. And from these 3 major nodes emit dozens of mitochondrial DNA variants, each at a relatively greater distance than the 3 major haplogroups. Obviously, since Babel, there have been many generations accounting for the relatively larger differences between the dozens of mitochondrial DNA variants we see today and the 3 major haplogroups from which they emerge. This is incredible and irrefutable. In a nutshell, if you're looking at the slide here, the picture, short lines equal pre-flood times, long lines equal post-flood times. Now, subnodes, well, this model makes testable predictions. If we come from these 3 nodes, and the DNA differences are all a marker of time, what we are looking at here with the radiating out from the 3 nodes are post-flood times or the history of civilization. We should be able to read the history of civilization from this tree. Romans, Greeks, Persians. Now the tips are individuals. Amazing work is actually currently being done on the transatlantic slave trade. Also, testable predictions have been made on the most divergent African peoples, the Khoisan peoples. Can evolutionists too make testable predictions on this map of humanity? Now, more on the entropic degeneration of our genome, as I think that's important here to the model, which in a nutshell is the fact that mutations are accumulating so quickly in genomes that natural selection cannot stop the functional degradation of the genome, let alone drive an evolutionary process that can turn pond scum to people. Think of rust slowly spreading throughout a car over time. Each little bit of rust akin to a single mutation in an organism is almost inconsequential on its own. But if the rusting process cannot be stopped, it will eventually destroy the car. Think of a book the size of, say, an encyclopedia, with each spelling mistake on its own is also inconsequential and does not seem to affect the book itself. But over time, as these spelling mistakes accumulate continuously, the book degrades and the message is completely lost. Most mutations are nearly neutral. They accumulate very small effects and accumulate relentlessly. All mutations must have some effect on fitness, no matter how small and trivial. There are obvious shelf lives on genomes, and that's what's important to take here. This is a long-term problem. Beneficials cannot counterbalance the accumulating damage. Due to the influx of deleterious mutations, the rare occurrence of beneficials cannot compensate for the information loss. Population geneticists acknowledge this very real problem. Obviously, mechanisms have been formulated, mutation count mechanism, synergistic epistasis, but all have been falsified. This is because low impact mutations continuously accumulate. There's a selection threshold. Genetic entropy in real time. Alzheimer's disease, dementia, autism, asthma, autoimmune disorders, cancers, immunological problems. There is a pandemic. Yes, this could also be epigenetic mutation, meaning at the end of the day, we are seeing entropic degeneration on all levels. We can increase selection intensity, but the problem won't be stopped. We are a dying people in a dying world. Our hope is in Christ. To put it into perspective, 700 billion mutations this generation, 100 per person per generation, 7 billion people on the planet, and most mutations are in the non-selection zone. H1N1 went from a red-hot pandemic to a whimper to an extinction event in 90 years. This was the human version. Genetic entropy, genetic degeneration at work. What about bacteria? Lenski is demonstrating reductive evolution, aka genetic degeneration. Reductive adaptation is consistent with genetic entropy. All beneficial mutations in Lenski's bacterial experiments are reductive: loss of function, loss of promoter, and loss of genes. They have painted themselves into a corner. They are lazy and handicapped. They are getting rid of genes short term, but it is long-term degeneration. We see genetic degeneration at all levels. It's so obvious and evident. Animals, humans, bacteria, viruses, it is irrefutable. It is recognized widely that if pawns come to people evolution occurs in the sense of universal common ancestry, single cell to multi-cell to fish to amphibian to reptile to mammal to bird to monkey to man, new novel information must be added to the genome. This information must be meaningful and it must be a net gain. This issue is a net gain versus a net loss. For a primitive cell to evolve into a whale, for example, the mutation/selection mechanism must increase the information from that in the cell until a whale's worth of information has been achieved. If you see here on the slide, we've got a few commonly presented examples by the evolutionists, but they're all insufficient because they are decay of prior information. That's not going to take your fish to fishermen. We got melanoprotein, nylon digestion, sickle cell anemia, aerobic citrate. There's many more, but at the end of the day, they're loss of specificity, loss of promoter, loss of genes, loss of information. Now, if we look at nested hierarchies, because we're looking at competing explanations. Now, if God designed life, what would we expect? What would we predict and assume he would do? God created us in his image. We somehow reflect something about the divine. Therefore, I think it's logical to assume and conclude that if we look at the way humans have designed things and the patterns that they have designed things themselves, we can get an idea as to how God designed life. Now, the
10-second warning.
10 more seconds?
That's right.
Now, if we actually go to transitional forms, we can see there's a competing explanation there. And we'll get into junk DNA later, but it seems like the trajectory is strong for genome-wide functionality. Genetics points to creation. Thank you so much, James.
You're welcome. So we will now go to Jackson Wheat for his opening statement. And we're very excited for Jackson Wheat's opening statement as well. Jackson Wheat has his links down in the description below, as well as Standing for Truths. So with
I'll
that,
get those to you afterwards. Sorry.
Oh, that's right. I'll get it right now. So I am resetting the clock and 15 minutes. The floor is yours. Thanks so much, Jackson.
Mind all the porn people. Okay. And all right, let's start. Whoops. Actually, let's not start. Technology. Gotta love it. Okay. Ready? Hello, everyone. Thank you to Modern Day Hysteria for having me on. Thank you very much. I appreciate it. Now, evolution is defined as change in allele frequencies in a population over generations, adding up to a record of natural common descent, and this proceeds via a number of mechanisms. Genetic mutations and recombination provide the substrate by which natural and sexual selection, as well as genetic drift and gene flow, act upon, eventually leading to speciation, which is macroevolution. This isn't merely inferred from fossils, but is indeed directly observed. Mutations, whether effectively or near neutral, harmful and beneficial, have been observed to occur in populations. As an example of a beneficial mutation, notothenoid icefish underwent a number of gene duplications, neo-functionalizing a duplicated trypsinogen gene into an antifreeze glycoprotein gene. This fact is ignored by all major creationist organizations. Natural and sexual selection, genetic drift, gene flow, and speciation have also all been observed and allow researchers to make testable predictions. Being able to make verifiable predictions is the hallmark of a good theory, and evolution has this in spades. Before we get there, though, I must point out that creationists accept all the mechanisms of evolution and most of what I've said so far, with the pretty much sole exception of beneficial mutations, even though nearly all researchers in the relevant fields do accept that these happen. The prevalence of mutations in organisms even allows researchers to make predictions about their prevalence in populations. For example, there are different types of mutations. For example, mutations from guanine to thymine, or mutations from cytosine to adenine, etc. These mutations occur at different rates in the human population, so are found at different frequencies. Using these frequencies, we can generate a graph like this, showing the signature of mutations. If mutations were also the cause of interspecies genetic differences, then we would expect to find a similar spectrum graph when coming up with all the different types of nucleotide differences between humans and chimps, and we do. In fact, the spectrum matches Also, when you look at the differences between humans and more distantly related apes, like gorillas and orangutans, and matches when comparing differences between those other apes, such as between chimps and gorillas. This makes no sense under a non-evolutionary model. Under creationism, it implies that God created interspecies DNA differences that just so happened to look exactly as though they had occurred by the same natural processes that give rise to within-species differences. It also makes no sense under a non-evolutionary model why genetic sequences for homologous proteins converge the further you go back in time. When we reconstruct ancestral sequences from different groups of species, we find that those ancestral sequences are more similar to one another than the descendant species sequences, implying a branching pattern of divergence that fits the evolutionary model of common descent perfectly. Nor does it make sense why we share specific pseudogenes, such as gulo, and specific endogenous retrovirus insertions in the exact same places in our genome as other organisms, or why we can make predictions about genetic events, such as the existence of a fusion site in chromosome 2, based purely on the idea of common ancestry with other organisms. We have also been able to make a number of predictions about fossils. First, Robert Broom predicted the existence of an amniote with a double hinged jaw joint based on the idea that mammals evolved from the colloquially called reptiles. This was discovered decades later in Proboscis and a whole host of other near mammal fossils. William Beebe predicted that birds should have gone through a stage in their evolution where they had asymmetrical flight feathers on their front and back legs, as indicated by embryology, which was found in the form of Microraptor. Also in relation to birds, a feather morphotype was predicted by embryological data and found in dinosaurs, such as Bipiosaurus. Paleontologists predicted an ant fossil that had features of wasps in the Cretaceous, and that was confirmed as Spheca myrma. Neil Shubin and colleagues predicted a fish-like tetrapod or tetrapod-like fish in Devonian strata of Canada, and that was confirmed as Tiktaalik. The list goes on, but the point is that there's no reason for these predictions to have been fulfilled if different clades of organisms were created separately from each other and flood geology were true. In some lineages, the embryos bear evidence of their evolutionary history. For example, fossils of terrestrial whale ancestors, such as Indohyus and Pachycetus, have the unique astragalus ankle bone of even-hoofed mammals, but also the unique inner ear of whales. In whale embryonic development, they sprout hind limbs that shortly recede into the body, but there's no reason for them to sprout these legs in the first place if whales didn't descend from terrestrial ancestors. This fact is so conclusive that it even caused baraminologist Kurt Wise to suggest that perhaps terrestrial whales had been on the ark. Whale embryos also start with nostrils at the tip of the snout, just like any other mammal, but during development, these nostrils migrate toward the top of the head. The migration of the nostrils is also observed in the fossil record, with early whales having the nostrils at the tip of the snout and later fossils showing the nostrils moving progressively toward the top of the head. This morphological transition is known as nasal drift, and it is well documented in paleontology. Under creationism, there's also no reason why baleen whales, along with all other toothless mammals and birds and reptiles like turtles, should have the broken remains of genes for building tooth enamel despite having no teeth. And there is no reason for whales to have hundreds of uniquely mammalian olfactory receptor genes, many of which are defective pseudogenes. In terrestrial mammals, these genes are involved in the sense of smell as they code for proteins that detect airborne chemicals, which isn't of much use to whales since they live underwater. Odontocetes, which are toothed whales, especially cannot use them since they have lost the part of the brain involved in the sense of smell altogether. But this makes perfect sense under the evolutionary model because, despite whales' adaptations to an aquatic life, they still bear all the stamps of their terrestrial mammalian ancestry, as well as specific genetic characters that make them more closely related to hippos and all other even-hoof mammals than anything else. In the case of whales, we have genetic, fossil, and embryological data all connecting whales to terrestrial mammals. This again makes no sense on the idea that they were created totally separately from anything else. Then there's biogeography. Geologists have worked out that the crust of the Earth has changed much throughout its history, and organisms have had to adapt to it. For example, notoungulates and marsupials independently converged on a number of forms similar to animals we're more familiar with, while they were separated from the rest of the world on South America. Speaking of marsupials, researchers correctly predicted that fossil marsupials would be found specifically in Eocene strata of Antarctica, since they moved from South America to Australia at a time when these landmasses were connected by Antarctica. Antarctica, like all other continents, has undergone numerous geographic and biological changes. During the Triassic, the continent had deserts that later became tropical forests, as indicated by pollen grains in the Cretaceous. Unless creationists are prepared to propose and provide evidence that all of Antarctica's, as well as all the world's flora and fauna, has changed several times over the course of the past 6,000 to 10,000 years, this data is not accounted for. In addition to this, creationists must account for the numerous lineages of organisms showing the acquisition of characters over geologic space. Creationists rarely, if ever, take note of the extremely gradualistic evolution of foraminifera, diatoms, and coccolithophores, and how these contribute to our understanding of biogeography and paleoclimatology. Or how pollen grains, spores, and various other paleontological objects inform us about ancient environments, or how all of these can be cross-dated by a number of independent radiometric methods. There is a mountain of data unambiguously indicating that the Earth is old, and creationists ignore almost all of it, heinously misrepresenting the little they do take note of. Now, what does the opposing side make of all of this? Evolutionary mechanisms make some changes, but there's no explanation as to where they end. Beneficial mutations, it is believed, don't happen, even though we have numerous examples. In Standing's case, he makes excuses for every single one. For example, he claims the apolipoprotein A1 Milano mutation caused a loss of specificity, hope you can hear the air quotes, in the protein, despite giving a family a selective benefit, an argument which was cribbed from Answers in Genesis. However, the way creationists use the word specificity, like information and kind and genetic entropy, doesn't really mean anything. In a way, we could describe all beneficial mutations as having lost some form of specificity. In the case of the notothenioid icefish, the duplicated trypsinogen gene lost its original specificity in becoming an antifreeze glycoprotein gene. But since the fish still retains its functional trypsinogen gene as well, this can only be considered a net gain. So it lost specificity even though it acquired a new beneficial function. Similarly, an enzyme that changes to now catalyze a second reaction at the cost of a reduction in efficiency in the first reaction could be described as a loss of specificity, even though it's obviously an example of the evolution of a new function. Creationists don't seem to understand that a large part of molecular evolution involves changing the existing activity of proteins or enzymes to produce new functions. Using deliberately vague and undefined terms allows anyone to spin whatever narrative they want and protect it by moving the goalpost constantly. Show me the evolution of new information or a new kind, creationists often ask. When you want to ask for a definition with clear, unambiguous criteria before you give an example, creationists will more often than not avoid answering the question or provide vague definitions for their vague terms. But when you do provide one example of new information or new kinds, creationists are suddenly quick to provide many criteria for why it doesn't count. And when provided with more examples, more criteria seem to appear out of nowhere. This seems a bit disingenuous on the creationist's part. Everything mentioned about the fossils is, in Standing's world, merely a happy coincidence. He barely touches the data, but then neither do the creationist sources he relies on, which is why most of his arguments are analogies. Analogies are great for explaining difficult concepts within an argument, but arguments still need some substance, which is ideally actual data or observations that are documented in the scientific literature. Creationists can't make fossil predictions like evolutionists do, because they have no basis for making such predictions, nor do they have any basis for making predictions about specific sequences of DNA among different, to them unrelated, groups of organisms. It's difficult to make any predictions when your model is literally based on magic and avoiding the vast majority of the known data. About the only prediction they could make is that there should be evidence of a population bottleneck among all terrestrial organisms about 4,000 years ago, which is a laughable failure. They have no idea which organisms are or aren't related to each other, since what constitutes a kind varies wildly, with no discernible criteria for determining kinds at all. And they have no explanation for why all of these different, independent lines of evidence arrive at the same conclusion, which is that all organisms share common ancestry. If Standing wants to demonstrate that creationism is true, he must account for all of this data. Putting forth one argument doesn't make any of the data disappear, and his position isn't viable until he accounts for said data. Thank you.
Excellent. So with that will we will now go to the rebuttals. So this is where it's going to start to heat up, get exciting, and I am excited. So one thing is we've got a lot of awesome questions already. I'm stoked to have them. And if you just so happen to have questions for Jackson as well, we will intersperse those, or you could say intertwine them with the questions that we have for Standing. Get ready, Standing. You have got a lot of questions, but don't worry. It's going to be an excellent time. So if you have questions for Jackson, we will usher those toward, like I said, not to the front of the line, but so that they'll be interspersed with the questions for Standing. And then Standing, very excited to have you go. And Jackson ceded roughly 2 minutes to be donated to the Q&A. And Standing for Truth, the floor is yours.
Awesome. Thanks so much. This should be fun. Jackson brought up some interesting stuff. This is probably going to get technical real quick. Since he had the notothenoid example up there for a while there, I'll touch on that first. The antifreeze protein, the glycoprotein gene that gets turned on, a lot of this is due to epigenetic regulation. For example, these fish, when they enter the cold water. Now, the thing to remember is that there are 5 families of the same gene. So the typical one, and Jackson's right, the SAS mutation gene that's found in zooarcid fish is a little different. It's mostly based on an amplification, but the notothenoid, I believe it's the trypsinogen gene that it mutates, and it's a different pathway, but still the same effect. So at the end of the day, it's just a epigenetic change. It's just non-random adaptation based on pre-existing heterozygosity. A big thing is phylogenetic systematics, especially if you're comparing these different sequences to different species and sequencing their genes, because phylogenetic systematics, it really just it's mostly based on the concept of outdated junk DNA, and because functions in DNA are only beginning to be found during the last decade, that really indicates that we only understand less than 5% of the DNA language, because the gold standard for testing for function would be gene knockout tests. And since we haven't done that on humans, it's premature to say that we understand the DNA language enough to know what's a mutation, what's functional, what's pseudogene. Now, if, as is suggested, say by the ENCODE, by many papers that suggested the vast majority of DNA is, a lot of it's preliminary, but it's pointing towards function. It takes away the primary assumption of phylogenetic systematics, which is based primarily on mutations and pseudo. So if the vast majority of DNA is functional, it becomes a common design argument, as we have always predicted as creationists. But we also know overall that the non-coding regions of DNA function much like an operating system in a computer. It's directing the timing, the expression, and regulation and use of the other information or the data files that code for proteins. So this regulation, this expression, these are significant roles. It also gives a clear, obvious identification that our genome is both polyfunctional and polyconstrained. So any change is going to be somewhat deleterious. It might not exhibit a clear phenotype, but it will be detrimental and deleterious. Now, another one he brought up was the melanoprotein, which it is a loss of specificity, because in gaining this antioxidant activity, the protein has lost a lot of activity for making HDL. So just like he said, I mean, the mutant protein has sacrificed specificity, but the fact that the persons with this mutation are unable to produce normal levels of HDLs, high density lipoproteins, which are known to perform a valuable role in moving bad cholesterol, suggests that there is going to be a health downside to this mutation, such as that is with sickle cell anemia. So a lot of these examples, for example, the notothenoid, it's just organismal genetic adaptation through environmental conditions, like all living creatures have based on pre-existing heterozygosity. Now, another one that he brought up was the chromosome 2 fusion. Just looking at my notes here. This alleged telomeric repeat site of the human chromosome 2 fusion that apparently, as Jackson expounded, corresponds to chimps' chromosomes 2A and 2B of, say, this hypothetical common ancestor, was actually a second promoter in this DDX2L2 gene. And genomic data for both the alleged fusion and cryptic centromeric sites actually refute the concept of fusion in a human chimpanzee common ancestor. So genetic data is actually refuting that. And even if it was a fusion, it would be indirect evidence, as it would have occurred in the human line. But genomic data, like I said, is refuting that. When it comes to the information net gain, that's because it really comes down to a net gain versus a net loss, because selection, it's going to remove the worst deleterious mutations and amplify the best beneficial mutations. But at the end of the day, a population can be undergoing genetic decline due to the vast numbers of slightly deleterious mutations, even while, say, selection may be amplifying a handful of beneficial mutations. But Jackson seems to fail to realize that, as I said, it can still undergo these minor adaptive fine-tuning to its environment, but it's still degenerating in many other ways. So natural selection it can certainly stop the degeneration problem, but it's not going to solve the degeneration problem. So when it comes down to net gain versus net loss, a few beneficial mutations are not going to counterbalance the influx of deleterious mutations. Now, a genetic marker that brought up was let me see here, was the gulo gene, and it has to do with vitamin C, and it was the loss of the vitamin C pathway due to deletions in the gulo gene. It's been detected in humans, apes, guinea pigs, mice, rats. Now, compared to chimpanzee and gorilla, though, the 28,000 plus base gulo region in humans is only 84% and 87% identical to chimpanzee and gorilla. The 13,000 base is upstream of the human gulo region, corresponding to the area of loss for at least two major exons, is only 68% and 73% identical to chimpanzee and gorilla. Also, the individual 6 exon regions, which are generally very similar among humans and various apes, each independently exhibit completely different and discordant phylogenetic patterns of similarity for all but one exon. So what I'm saying is a general lack of human SNPs in these exons indicates that incomplete lineage sorting based on alleged ancestral polymorphisms is not likely an explanation. Now, there's a lot of inconsistencies with the genetic markers that are being used, especially because we are now seeing that there are many known functions. Now, if common ancestry is true, as he's saying, he talked a lot about convergent evolution, but that certainly doesn't help the case for pawns come to people, because why do whales have a water echolocation system lacking in land mammals? And if they came from land mammals, which contains the same smelling gene, why are whale echolocation genetic markers generally absent in land mammals? Now, another thing he talked about here are endogenous retroviruses, which we know are playing important roles in the embryo, immune response roles as well. I think it was in, let me see here, he talked about the reptile to mammals, say therapsids. It's not as consistent as he talks about. Fossils more mammal-like can occur earlier in the fossil record than fossils that are less mammal-like. So yet to trace an evolutionary lineage on the basis of the fossil record requires that therapsids structurally more similar to mammals enter the history of life later than those that are structurally less similar. So people evolution, after all, universal common ancestry, needs to follow time's arrow and cannot have offspring giving birth to parents. Now, A similar problem in regards to this arises with geographical mismatch, in which fossil organisms that are supposedly next to each other in a structural progression are actually widely separated geographically. And Jackson might be able to admit to that. But if the geographical separation is too great, how can one organism be ancestral to the other? Reproduction, after all, requires proximity. Parents do not give birth to offspring at the other side of the globe. I think that's clear.
30-second warning.
Like I said, is a huge problem for these phylogenetic trees. And also in regards to the reptile mammal, let's look at the evolution of the mammalian ear from the reptilian jaw. How exactly did those two bones from the reptilian jaw migrate to the mammalian ear? Now, the word migrate in this context is empty of scientific content. What genetic changes and selection pressures were in fact operating, and how specifically did they bring about the evolutionary pathway in question? I talked about the Evo groups. That's
the rebuttal from Standing for Truth. Excellent so far. Very articulated responses from both debaters, or I should say statements. And so now we'll go to the first response. This is from Jackson Wheat, who's going to give his first rebuttal. So very excited to have this, and resetting the timer. So Jackson Wheat, the floor is yours.
There was an incredible amount wrong with Standing's rebuttal, but I'm afraid I'm going to have to save that for my closing statement because I prepared my rebuttal in advance. I knew that Standing would, be unable to counter most, if any, of the points that I had, so I wrote this in advance. So, before we get into the argument itself, it's important to note that the idea that organisms are undergoing genomic degradation over generations was held by creationists even before there was data that could actually be used to confirm or refute this position. I have a book from 1967 titled Did Man Get Here by Evolution or by Creation that asserts organisms are breaking down over time, never gaining new functions or structures. Therefore, Standings is, at its core, a doctrinal position that he and other creationists have been scrambling around to provide data for half a century, rather than honestly evaluating the data as it comes in. Now let's get to that data. First, what is meant by near neutral? Well, mutations can be near neutral and be either harmful or beneficial. A beneficial mutation is a mutation that provides a selective benefit to the host, and this definition will be important as we move forward. According to the 2007 paper, The Distribution of Fitness Effects of New Mutations, quote, all mutations must have some effect, even if that effect is vanishingly small, close quote. The paper goes on to say that those mutations with vanishingly small effects are called effectively neutral because they do not have any noticeable impact on the host. In Sanford's argument, near and effectively neutral and harmful mutations are expected to accrue almost nonstop in a population because natural selection is too slow to weed them out. This, in turn, relies on a complete mischaracterization of Moto Kimura's neutral theory of molecular evolution. In that theory, Kimura allows for the vast majority of mutations to be effectively or near neutral or harmful. However, beneficial mutations overwhelmingly overpower the former types. In his own words, quote, whether such a small rate of deterioration in fitness constitutes a threat to the survival and welfare of the species, not to the individual, is a moot point, but this will easily be taken care of by adaptive gene substitutions that must occur from time to time, say once every few hundred generations, close quote. Sanford was therefore lying when he said, quote, population geneticists know that essentially all mutations are deleterious, and that mutations having positive effects on fitness are so rare as to be excluded from such distribution diagrams, close quote. And anyone who had checked Sanford's claims could have found that out. With that in mind, the question becomes whether advantageous mutations do indeed exist, and the answer is overwhelmingly yes. Let's take some examples. For starters, garter snakes have repeatedly developed resistance to tetrodotoxins in their prey, the rough-skinned newt. Second, the water strider genus Ragavalia had a gene duplication that allowed the formation of novel propeller fans that gave it access to a new environment. Third, the Arctic cod, Boreogatus, actually turned a non-coding region of DNA into an antifreeze glycoprotein sequence, again, utterly ignored by creationists. Now, we could go on for hours talking about beneficial mutations, but the point has been made. There are numerous documented examples of organisms acquiring mutations that help them better survive in their environment. So how does Standing respond to this? His argument in the past has been that all of these are irrelevant unless we can show that hundreds or thousands of mutations in every species, which isn't even required by the model Sanford is misunderstanding. In essence, Standing is moving the goalpost to the moon. Sanford also misrepresented James Crow's work when Sanford claimed Crow said that all populations are slowly declining in fitness. Crow's work was only applied in that case to humans who are not under any strong selective pressures. This is the exact opposite of what Sanford is trying to argue, which is that natural selection is too slow to counteract the effects of deleterious mutations. Another example of misrepresenting the works of scientists, Sanford added a graph in his book with the name, quote, actual biological data showing the mutation accumulation and fitness decline in the human influenza virus, close quote, to support his claim on genetic entropy. As Sanford describes himself, quote, this graph illustrates the pathogenicity, i.e. fitness, of the H1N1 strain as well as the other two pandemic-causing strains during the last century, close quote. Sanford references figure 1 from the paper by Simonson et al. For this specific graph, but the original paper doesn't say what Sanford claims it says. As Gerard Jellison points out in his review on Amazon, quote, the point of the paper is that the flu epidemic of 1918 to 1919 was unusual in that it killed mostly young people. The author studied how this ratio varied with time and modeled the results in terms of different immunity in the two age cohorts. Thus, the curves in Sanford's plot merely show that, as time goes on, flu strains kill a greater proportion of elderly people. This makes sense, since young people have more vigorous immune systems. Sanford's figure is invalid since it does not illustrate declining viral fitness at all. This blunder is an absurd misuse and or misunderstanding of scientific data, close quote. We can also apply Sanford's model to paleogenomics, which doesn't in any way support his case. For example, paleogenomic and paleoproteomic studies have been done on mammoths, mastodons, glyptodonts, the American cheetah, saber-toothed cats, giant sloths, toxodon, macrauchenia, stilt-legged horses, Neanderthals, Denisovans, archaic humans, and various other organisms. And yet there is no sign whatsoever of increasing genomic degradation as time goes on. In summation, Sanford, and therefore Standing, has utterly failed to provide evidence for his genetic entropy model, as it isn't supported in modern humans, paleogenomic samples, long-term bacteria experiments, and yes, I will have data in the description for that one, or even the H1N1 model that he attempted to apply it to. Now, for a few notes on created heterozygosity. This is essentially the idea that Adam and Eve were created with extremely high heterozygosity, as in many positions in the genome were different. And this variation was all recombined to account for the genetic variation we see in modern human populations. Now, in principle, this is possible. Two individuals with miraculously high heterozygosity could give rise to a modern population with comparable genetic variation to what we see today. But then again, it shouldn't be surprising that you can make things like this work by invoking a magical starting point. However, while this scenario could, in principle, account for the observed human genetic diversity, it doesn't. Analyses that basically track back all the recombination events that Jensen requires to see when the alleles could have been present in just two individuals have shown that those two individuals would have lived at least 400,000 years ago, given observed mutation rates and recombination rates, so it doesn't fit a YEC timeline. In other words, created heterozygosity more recently than 400,000 years ago doesn't match the data. This is in addition to the fact that it's a completely ad hoc rescuing device. Basically, creationists noticed that there was too much genetic diversity and said, well, all those nucleotide differences that couldn't have been caused by mutations in the last few thousand years, yeah, those are magically created by God. In effect, Jensen is saying God specially created humans with all the variation they would have had if they descended from an earlier population in the distant past, but they didn't. Jensen all but admits it's completely ad hoc, but says it's fine because evolutionists do the same thing. Humans contain more genetic variation than could be the result of mutations in the past couple of 100,000 years too, implying that the ancestors of modern humans also had heterozygous genomes. Jensen essentially says, look, we both require heterozygosity in our ancestors, so we're on similar footing. The obvious problem is that Jensen's invocation of created heterozygosity is entirely ad hoc, while evolutionist invocation of pre-existing heterozygosity is absolutely not. According to evolution, populations are what evolve, meaning there will always be genetic variation in an ancestral population, since that ancestral population have their own evolutionary history. Some of that variation will be inherited by the descendant population, meaning that new species will contain more genetic variation. Evolution expected pre-existing heterozygosity and found it when the first data came in. Creationism doesn't inherently predict pre-existing heterozygosity and only invokes it to account for the inconvenient data after the fact. All of this, of course, is in addition to the overwhelming evidence we already have of ancient timescales, human evolutions, and mutations being responsible for both within human genetic variation and between species differences, such as the mutation spectrum we talked about before in my opening statement. Now I want to move on to a few things that Standing said in the time that I have. One of the most ridiculous things that he said was regarding the whales. He said, oh, well, land mammals don't have the genes for echolocation. Duh, they don't need them. Obviously. He talked about linkage groups, and I wasn't really understanding where he was coming from. Maybe you can explain that to me better. But linkage groups are basically just the genes found on a single chromosome. So I don't really understand where he was going with that one. Haplogroups M and N and L were not all simultaneous. M and N came from L, right? One came before the others. You said evolution doesn't explain. Oh, sorry. Well, I'll cover that one later. It needs a longer explanation. The chromosome 2 thing, you said, you talked about DDX11L2. It's a telomere-specific pseudogene, so it's no wonder why it's found in the center of this chromosome arm, which even Tompkins in his own research depicted as looking like a telomere. It's not a wonder. I did a full analysis with the help of
15 seconds left.
How much time?
About 15 seconds left.
Okay, well, I did a whole analysis with Rational Mind and Nestlig. I'm sorry, Nestlig, on that, and so you guys can check that out. But I'll end that for now and get to the rest later.
You betcha. So very clear rebuttal from our good friend Jackson Wheat. And so I appreciate the debaters, by the way. Both of them make me think this, but Jackson, especially as we've had our exchanges, is that I appreciate that both debaters are just so civil. And so it's been a good time. So next, we are going to move to our 5-minute responses. So these are short and sweet. And Standing for Truth, excited to hear from you. So the floor is yours when you are ready, buddy.
Actual real-time evidence for genomic degradation, which is weird because I use the, for one, the H1N1 virus that went to complete extinction in humans. Also, Lenski's experiment, which what's funny is Richard Dawkins in his book, Greatest Show on Earth, presents the E. coli bacterial experiment as the #1 best proof for evolution. But what's funny is that experiment on the E. coli bacteria actually support genomic degradation, because although an adaptation happened, the bacteria have shrank in genome size, and the more rapid growth that was observed was actually accomplished through genetic degeneration, as I talked about in more detail in my opening. And many useful genes that are not essential in that environment, it's an artificial environment, they were lost. So transfer these already degenerated, these handicapped, these lazy bacteria to a natural environment. Those highly degenerated and handicapped bacteria will most certainly be dead on arrival. Also, we see plenty of evidence for genomic degradation in Neanderthals. We see it in, I think it's Homo erectus, Homo naledi, the hobbits. We see clearly that there were inbreeding going on, and there were high levels of homozygosity, reductive evolution found in this, which kind of explains the weird degenerate features found in some of these ancient species. Also, when it comes to genomic degradation, population geneticists do acknowledge that this is a real problem. I mean, we're inheriting these deleterious mutations, and it really is about net gain versus net loss, because a few beneficial mutations will not counterbalance the accumulating damage. This is a fact. And if all known examples are degenerate in some way, loss or decay of prior information, or just general adaptations, say found in the notothenioid fish, then if you're taking one step forward but 10 steps backwards, it's not going to help your pond scum to people evolution. And these population geneticists have come up with mechanisms such as mutation count mechanism, which pretty much states that as mutations accumulate, Mother Nature somehow counts the number of mutations per person, and then Mother Nature selects the higher number of individuals. But simulations have disproven this, and it's not biologically real. So that's insufficient to explain the genomic degradation problem. But most importantly about genetic entropy is it's consistent with the biblical model after the fall. We know that the Bible records a time of longevity, long lives, and a huge decay curve is seen, the same decay curve that we can see based on this accumulation of deleterious mutations. Another one is synergistic epistasis, which pretty much says that if we have lots of mutations that are deleterious, as they accumulate, they amplify each other's deleterious effects. The intensity of degeneration is so great that somehow it improves selection, but it's now demonstrated that introducing this synergistic epistasis only makes the problem worse. And that's actually what logic would dictate. So there's plenty of real examples of genomic degradation. I think you might have just ignored them all. The biggest praised example of E. coli is demonstrating genomic degradation, H1N1, like I said, Naledi, Erectus, Neanderthal inbreeding, reductive evolution related populations. Just look at all the degeneration going on, the mutation, diseases based on mutations, whether it's the autoimmune diseases, cancers, entropic degeneration on all levels. It's a fact. Now, he also, I didn't touch it before, with gene duplications. And at the end of the day, if you're duplicating an already degenerate gene, you're only speeding up the degeneration process. So it's still not going to answer the question of net loss versus net gain. And also, it looks to me like the genetic markers used to prove, say, pond scum to people evolution through phylogenetics, they seem to be cherry-picked data. Like, for example, the insulin peptide gene, it really does look like a lot of cherry picking going on because this is only a very small subset and it's very, very incomplete data. This is a very small sample. I think it's only 330 nucleotides. So this pattern is representative of a nested hierarchy according to evolution, but with it being only a small subset, it's really not evidence for universal common ancestry. And convergent evolution, when invoked, when there's an inconsistency in the data, is evidence against this apparent tree of life.
10-second warning.
So I would say with the pre-existing heterozygosity, we know 850 horse breeds have been derived just from little DNA differences. I mean, I think the model's strong. Genetic entropy supports creation. It's evidence against evolution. So I'm pretty happy with everything I've said here. Thank you.
Okay, so excited to have our final closing or response and then move into the Q&A as we have a lot of questions. We had to close the Q&A a little bit early for the intake as we have a ton of great questions. So I'm resetting the timer, and Jackson, the floor is yours. Excited to hear you.
Whoops, there we go. I'm sorry, we pressed it at the same time. Okay, so Standing, I feel like you completely ignored my rebuttal. And also, for example, I specifically noted the H1N1 virus and how Sanford misused the data. He misused the data, and other researchers can see that he misused the data and called him out on that. And you literally just ignored it. It went over your head. I am shocked at that. Oh my goodness. I feel like I already covered the chromosome 2 thing. This is really, the chromosome 2 argument is really not even argued by like 98% of the field. So whatever. I already mentioned that it's a telomere specific DDX11L2 pseudogene. You said it has function. No, it doesn't. It's a pseudogene with no known function. Actually, a lot of research has gone into it, and it has no known function. You mentioned the Linsky E. coli experiment. Actually, research has shown on that that series of experiments that the upper bound, there is no upper bound to the fitness on the bacteria. They're not just degrading. Their fitness has no upper bound, right? So you claiming that is just completely without data. You completely misrepresented the notothenioid icefish example. No. I know you tried pulling over to Zoarcid fish, which I think is a little funny, and I'll get to that in a second as to why. But no, you have an example of, it's called neofunctionalization because it came from a gene that had a totally unrelated function. Now, what's being used here is it's what is it? Threonine, alanine, the amino acid, or which is the repeats that make up the antifreeze protein. And so that's being used. It had this other job which was not involved in antifreeze. It was involved in trypsinogen, but it came to be used for this other thing, this completely new function after it was duplicated and mutated a number of times. You pulled over to Zoarcid, which is interesting because that's what Georgia Purdom did in her chapter on the Answers book, which I read, because she data suppresses. She doesn't note the fact, she does point out, yes, there are 5 different kinds of antifreeze genes, but we're only going to talk about the one that's really an amplification of something that's already doing sort of antifreeze. Data suppression. Wow, what a surprise. I mentioned the Boreogatus ice fish also, which you didn't even mention. It took a sequence of DNA that had no coding function whatsoever, and it turned it into an antifreeze protein. So again, ignored by creationists. You mentioned common design earlier. You said, "Well, why isn't this similarities examples of or evidence of common design?" Well, common design makes no predictions, none whatsoever. Also, how would you explain the differences on common design? Would you say there are different designers for every difference? How would that work? I pointed out specific examples of things that we should find if life shares common ancestry, such as the homologous sequences of proteins. We should find that they converge the further you go back in time. What a surprise. That's exactly what we find. The apolipoprotein A1 mulatto mutation is beneficial. You don't seem to really understand that there are trade-offs in evolution. Evolution is all about trade-offs. You don't always get this net gain, but trying to say, oh, well, it doesn't matter because not necessarily net gain. You don't always get everything, 100% gain new thing. A lot of times there are trade-offs involved, which I mentioned in my opening, which seems to have been ignored. And so in this case, you have a trade-off. It's still beneficial. So at the end of the day, it's still a beneficial mutation. It's not causing them more harm than good. Oops, sorry. You mentioned, you dropped that ERVs play important roles in the embryo, like one or maybe a few. There are lots of ERVs. Not all of them play roles in the embryo, right? So you just kind of dropped that there and ran, but that's not the case. You kind of seem to lack an understanding of how phylogenies are constructed when dealing with fossils. If you find, like, you made a claim that, there are more advanced mammal-like critters found before the more primitive ones, such as, you can find more advanced things before more primitive critters in the fossil record because the fossil record is very scattershot. We have an understanding of how taphonomy works and how fossilization works, and so we understand not everything's going to get fossilized, and so we're going to get a picture, and then we have to ask ourselves, well, is this an accurate picture of what happened? There's a whole discussion on taphonomy and paleontology we could have, but this isn't the time for it. How did the jawbones migrate to the ear? Well, actually, the genetic work has actually been done on that. There's a lot of interesting genetic papers regarding that. Researchers can kind of reverse engineer the jaw and the teeth to kind of see how that worked.
10-second warning.
Dang. The paleogenomic thing you said was complete nonsense. There's no evidence of genomic degradation in Homo. And when you said cherry picking, that's nonsense for how determining phylogenies. We look at millions of nucleotide sequences. So nonsense again.
So we will now go to the Q&A. We have a lot of great questions. I'm excited to read these. So we have a pretty good mix now. So we had a number of people where we could kind of intersperse questions for Jackson into these questions for Standing for Truth. So Super Not Bad asks Standing for Truth how young Earth creation deals with distant starlight in a young universe.
Jackson 4, it looks like Super Not Bad gives me another tough question to answer. You can certainly go with the supernatural explanation. I mean, obviously, God created a full-grown Adam, full-grown garden, full-grown mature Earth. If scientists were to come and do their blood tests on Adam, who say was created just an hour before, you know what I mean, based on height or weight, they might conclude that he's old. Based on blood tests, cholesterol levels, they might conclude that he's young. So at the end of the day, looking at starlight, if God stretched out the heavens, as the Bible says, you can certainly go that route. I prefer to go the more scientific route. Dr. Humphreys, gravitational time dilation. You'd have to go through a lot of reading material to understand. It's kind of hard to go in a 1-minute answer, but if we assume the Earth is, say, at the center of mass in space, something in the center of mass will create a huge dent in space. Okay, now we would also have to assume that God used what's called a white hole to bring about the universe. Now, given the two scenarios, if you're following me, the Earth is in a huge gravitational well, and deep inside a gravitational well where time slows down. Okay, so here's the explanation. And the deeper you get, the slower time gets to the point where time itself stops. Now, while the Earth is inside that well, God also created stars and galaxies inside that well. This is where it gets interesting. So the Earth, and I'm trying to put this into a nutshell. So the Earth and the stars are inside the well at the same time. And that well is a white hole, like I said earlier. But while God was putting stars and galaxies inside that well, it causes the original dent in space-time to increase further, sending the Earth down much further into that well, causing time to completely stop. I know it's kind of technical, but it does explain the distant light problem. Now, while Earth clocks were stopped and everything from stars and galaxies was inside that white hole, just like the Bible said, God stretched out the heavens, which means, I think it's pretty obvious, he expanded the event horizon of the universe starting from the edge of that white hole. So when he stretched it out, stars and galaxies were the first to come out of that well. And when you escape that gravitational well, time can now start ticking. So which clock started ticking first, right? Stars and galaxies or deep space? I'm almost done here. I just want the audience to understand that we do have a strong model, because starlight started traveling from the event horizon to the center of mass, which is the white hole. Now, as God continued to stretch out the universe, the Earth finally was pulled out of that white hole, and for the first time, Earth clock started to tick. When Earth clock started ticking, since galaxies and stars came out of that well first and had billions of years of cosmic activity, while the Earth was still in that well, light from those galaxies already traveled to the center of mass, and when the Earth came out of it, light distant starlight was already there. So I explained that in a nutshell. I'm very interested in this distant starlight problem. That's the best I can explain in the limited time I have, but I would definitely suggest people look. You can go at the supernatural explanation, or you can go for this really technical explanation. Dr. Humphreys, I recommend looking into it. It completely solves the problem.
So excellent. That was a good question and a long answer, and not to say it long.
It's kind of a hard question to not give a. But
yeah,
no, I know what you mean.
No, that's fair. I agree with that. I think you're right. Like, I didn't mean to like undermine it anyway. So
no problem.
What we have next is a question for Jackson, and this came from Dragnoct Silvas. He asks, how do scientists determine haplogroups for extant and extinct individuals?
Well, haplogroups are, so in your mitochondria has its own DNA, and so, which is called mtDNA, and so it has these groups has these sequences of nucleotides, and these are specific sequences. They call them haplogroups, and so when you look at populations, you can see how these sequences of DNA mutate according to whatever the biogeographic region is, or whatever the geographic region is, and so you can kind of show the biology and the geography. It's really interesting. But I mean, how would you determine them? I guess by sequencing their genome, their mitochondrial genome.
You bet. Excellent. So definitely a tough question. That was one where I was like, I don't understand more than half of the words. So that's excellent. Next, we have a question from Sabella Shepherd, who asks, Ask him why he's, let's see. Okay, this must be for Standing. They said, ask him or Standing why he's leaving out one very important person in the creation story. Not sure who they're referring to. Do you, Standing, you have an idea of who that might be referring to? And they put creation in quotes. So they said,
I can't see any of that. That chat was moving. I can't see any of the, did she put a name in there or something?
no.
Am I leaving out somebody in the creation account? Is she talking about Adam, Eve, and
maybe
who's that? Apparently a weird. It's a Jewish fable. It has to do with Adam had a wife before Eve or something. Is that what she's talking about?
Yeah, I'm not sure. It says ask
Lydia,
why.
maybe.
I just don't know. So we might have to come back to that one. And we do have another one though. So we have for Standing. Dragnoct Silvis asks, what is the function, in quotes, quote unquote, function of genes within gene deserts?
So I think based on the fact, and as I explained earlier, we only speak 5% of the genome, if that, based on the gold standard of genome function. And based on pre-existing heterozygosity, our prediction, if you were following, involves the non-coding DNA regions. And evolution says there's going to be much non-functionality, say maybe 20%, while us as creationists predict functionality of most of the genome. And I would say the results from ENCODE have suggested that there's a strong trajectory for that genome-wide functionality. So in regards to the gene deserts, I mean, it's hard to say exactly what the function could be until we've actually done the necessary functional tests. But I would say that the non-coding regions, or technically the regions in general of the DNA, like I said before, do function much like an operating system in a computer. So whether it's expression, regulation, timing, it's going to have, I might not be able to tell you exactly what the use is now, but with further research and the way things are going, I mean, back in the 40s, it was known one gene, one protein, one function. So many things have been overturned. I mean, RNA was once underappreciated. We know it's doing so many unexpected things. So you know what, in a few years or with further research, I might be able to tell you exactly what it's doing. But a lot of these results are preliminary. So I would just say the trajectory is clear. We're looking to more and more function, but I can't exactly tell you what all the specific functions are, especially if we haven't tested the developmental stages, because those were not evaluated in ENCODE. Only systematic knockout experiments could demonstrate that. So that'd be my answer.
Gotcha. So next we have a question for Jackson. So we got two in a row. We're going to come back to that one because somebody in the comments actually pointed out that it was Lilith that they were.
Yeah.
But
and that's what I thought. We can go to Jackson, and then I'll answer that after.
Okay, so William Estes asks, this is for Jackson Wheat. Some Christians are afraid to admit to evolution. Any advice on how to accept the theory without abandoning the faith?
Well, simple. Point out that there are lots of in fact, there are more Christians who accept evolution than there are Christian creationists. That's an important point, I think, to point out. Look at all of the wonderful work that Christians who are evolutionists have done. Look at Francis Collins, who is the head of the Human Genome Project. Point out that it's not, or Robert T. Bacher, who is a paleontologist and also a Presbyterian minister. Point out that you don't have to abandon any of the tenets of Christianity to accept evolution, and the Christians don't. The Christians who accept evolution don't do it. They don't reject Jesus or God or anything like that. And so, I understand. I'm in Louisiana. I understand being tentative about wanting to come out with such information. But I think it is important to point out that these two are not, that Christianity and evolution or science generally are not at odds with each other, and they shouldn't be construed that way. Those are my thoughts.
You bet. Gotcha. So thank you for that. And next we have, so we'll go back to that initial question from the person named Sabella Shepherd, who asked, this is asking for standing, why he's leaving out one very important person, Lilith, in the creation story.
Well, I think even to theistic evolutionists, old Earth creationists, obviously young Earth creationists, none of us are going to believe that Lilith was part of the creation story. It's based on fables. It's based on extra biblical evidence. I mean, if you look at something like the Book of Enoch, you know, like obviously somebody decided to take up the opportunity to write a book, because we know Enoch is mentioned in the Bible. I think it's Jude where it's mentioned that, you know, there was a Book of Enoch. And at the end of the day, you look at something like that and you see how ridiculous it is. But for the people that take that book, say the Book of Enoch, and actually get their doctrine from it, is the same type of way and formula that a lot of people have developed their doctrinal beliefs on Lilith. So, I mean, it's found nowhere in the Bible. Like I said, I don't think even theistic evolutionists are going to agree that there was a Lilith. I think in regard to the Bible, I think it's pretty clear that Adam and Eve were Jesus himself said that at the beginning of creation, he made them male and female. And I know a lot of people try and say, oh, that's out of context. Context is divorce. Well, obviously he's talking about divorce, but he's talking about that was instituted at the beginning of creation, which would have been the creation of Adam and Eve. So I don't see anywhere where the Bible talks about anybody named Lilith. I think it's based on fables. It's based on extra biblical evidence. And to be honest with you, I think it's certainly fiction based. So that'd be my answer to that. I'm not sure if you're familiar with Lilith, James, but that's really all I got for that.
It is new to me, to be honest. However, interesting to learn about. So I am going to go to your next question, if you're all right with that, because I know that we've got a number of them for you, and I want to kind of get back to the next one so we can try to squeeze these in. So Biblical History Skeptics asks, how does standing cope with the fact that the Bible has numerous different creation myths, including one where God creates via killing chaos dragons?
So I think from what I know, a lot of people try and use Genesis 1, which talks about the 6 days of creation, and then how it goes into Genesis 2, and it kind of zooms in on the bigger picture of the creation of Adam and Eve and what happened with that, and how Eve was created out of Adam's rib. So a lot of people will say, oh, there's a contradiction because there's 2 creation accounts, but it's actually just one creation account. I mean, hermeneutically, reading through that and exegeting both Genesis 1 and 2, you can clearly see that it's just a general overview in chapter 1, and chapter 2 is just a detailed description of what's actually happening. I think it's on day 6 with Adam and Eve. So I don't know why evolutionists still use that. In regards to the dragons, I'm not sure if he's talking about the Leviathans. He'd need more clarification on that. I'm not too sure what he's talking about there, but the only objection I've heard was, hey, Genesis 1 and 2 are contradictory because they're 2 different creation accounts. But I think it's pretty obvious to anybody who's reading the Bible and using proper hermeneutics that it's just it's the same creation account. It's one creation account.
Thank you very much for your response. And next we have a question from William Estes, who asks, standing for truth, can you compare the known genetic bottleneck in cheetahs with whatever evidence there is for a flood bottleneck? So
a lot of my evidence for the flood bottleneck was situated in the 3 major haplogroups, because the L, M, and N are close together. And I think Jackson Weed actually kind of used a straw man during his debate. I didn't have much time to rebut it, but the N and M are connected to L. That's true, but all 3 are close together. So that's the entire point. The other L's are farther than the primary L, M, and N. But the fact that there's 3 major haplogroups, and we know that after the ark, the flood bottleneck, mitochondrial DNA is inherited exclusively through women. So Noah's 3 sons wouldn't have passed that on, but Noah's 3 sons' wives would have passed that on. And what we see is 3 major haplogroups. I mean, that's completely consistent with biblical creation. I was even talking to an atheist the other day about that, and you just said, well, couldn't that be coincidence? Well, at the end of the day, they can use a rescue device of hey, that's a coincidence, but that's a pretty big coincidence. And if you're a biblical creationist, you should look at that proudly, and you should say to yourself, wow, the empirical scientific data points to, for one, the flood bottleneck with the 3 major haplogroups. When it comes to the cheetahs, very little heterozygosity and genetic diversity would have been lost because the bottleneck was so short. So if we were to, I talk about shifts from heterozygosity to homozygosity would lead to, new species. You'd have to have migration as well. Radiation from the flood definitely would have sped up that speciation process. But if we genetically sequence, say, the genomes of cheetahs and see what levels of homozygosity there are, versus, say, other species, we might be able to look at, like, for example, Dr. Nathaniel Jensen just wrote a paper about the finches and how there were shifts from heterozygosity to homozygosity leading to new species. So that's a perfect real-time empirical evidence of our model of created heterozygosity. So that's really all I got for that one.
Our next question, thanks for your response, by the way. And our next question comes from Landon Freeman, who asks Jackson, what would the requirements be for neo-Darwinism, and then in parentheses puts, not change over time in general, to be falsified?
I think that's a great question. That's one I've been thinking about. And I think one of the best examples for that is to not have increasingly inclusive groups of organisms. Like, for example, if every single kind, let's say, had, or if you had different groups of organisms, each of them who had completely separate genetic material that it could be shown biologically, chemically, whatever, could not go from one to the other, that would falsify evolution, for one thing. We can make tests based on creationism, and if the predictions that we have made didn't come true, like, for instance, if we predicted that that homologous sequences of proteins would converge as we went back in time, if we went back and checked, no, they're actually all different from each other, they're nothing like each other, then that would falsify evolution. If we compared, for instance, the human genome and the chimp genome and the genome of the pufferfish, and it showed, well, actually, there are only 2% differences in the genome between us and the pufferfish, but 95% differences between us and the chimpanzee. Well, that would falsify the idea of connecting homology and genetics, because it wouldn't make any sense that we would both share a close common ancestor when we have all these similar features if the genetic code is totally different. So yeah, I showed a number of models that we can make predicated on the idea that we share common ancestry and that all organisms share common ancestry, and they are precisely what we find. But it is a very good question. So,
you bet. Excellent. So we have, let's see, just a little bit more time left, and Jackson, you asked if you could give a response to the suggestion by Standing that you made a straw man. And so if we give you just like maybe a minute to clarify what your intention was in that when you did put your argument forth, is that fair for you, Standing for Truth? If we give him a minute, if you want, we can give you a minute, but then we got to return to the Q&A. I think maybe how about this? If we give Jackson a minute to respond to this one, then Standing for Truth, if he wants to respond to a question in the future, he can give a 1-minute response. Is that okay? I think my volume went out for a second. Sorry.
Standing, your mic is off.
If I give Jackson 1 minute to respond to you, Standing, for this, and then Standing, in the future, if there's a question that you want to give a quick 1 minute response to keep it even, is that okay with you?
I think that's completely fine. I think it's only fair. I was wondering if he gives a 1 minute response to what I said about the straw man, can I take literally 20 seconds to address something that he said that I failed to answer? It would just take me 20 seconds. Would that be fair? Or?
Is that okay, Jackson?
I mean, I can say,
It has to do with the H. Okay, just you can go ahead, Jackson.
I mean, this is going to get too complicated. I can say it doesn't matter, and we can move on. It's not going to kill me. All right. Besides, Standing and I have each other's email, so we can always talk about it afterward.
Okay.
It's not going to kill me.
True.
Okay, that's fair. Sorry, I just know that you guys are both passionate, and so I'm like, I want to treat you guys as fairly as possible, given that you're both passionate and passionate.
And you are.
Huh?
And you are, brother. You're doing great.
Thank you. I appreciate that's nice of you to say. I appreciate it, man. Let's see. For our next question, we're asking, this is from Old Scratch, asks, does Standing have any peer-reviewed scientific research that supports his conclusions?
Oh, yes, there's plenty of peer-reviewed sources. Actually, a paper was recently written on the limits of mutation selection. Obviously, the second that I mentioned that if it's Dr. John Sanford, they're going to have a problem with that. But this is peer-reviewed a rock solid paper. It's kind of related to Haldane's dilemma. Just to add 2 new nucleotides and actually, have this fixation in a population, beneficial mutations leading to new species. The amount of time that would be required is beyond any time that evolutionists would even believe has existed. And it's a rock solid paper. I can actually post it for James to post as well. Peer-reviewed sources, especially in regards to genomic degradation. I mean, you can read the Lenski paper yourself. You can read all the different papers about all the mutation-related diseases. In regards to the Darwin's finches and how there was a shift from heterozygosity to homozygosity leading to these new species. You can certainly read the original paper to that. That was the conclusion. And the thing is, creation scientists, they'll look at a lot of these peer-reviewed papers as well. They have a peer-reviewed team as well, so they're doing their due diligence, but they'll come to a different conclusion because there's a different worldview. So I notice a lot of people like RJ, he may think that, someone like John Sanford is leaving out data, or he's not coming to the same conclusion that the evolutionist did, but of course he's not. He's looking at the data and he's seeing how consistent it is with genetic degeneration on all levels, and based on his worldview and based on our worldview, that's consistent with the empirical data. So we don't always have to come to the same conclusion as the evolutionist does, but there's tons of peer-reviewed papers.
Gotcha. So we will go now to another question. This one's for Jackson. So Jackson, Jesus is Lord asks, can you explain how scales transformed into feathers for flight? Was it air friction?
No, I don't even know was it air friction? I don't know what that means. But as it happens, there are actually some really nice Prum and Brush papers in the 90s and 2000s, even to today, and that explain how you go from scales to feathers. You basically have you have the plumulaceous feathers and the pinnaceous feathers, and you can see the step by step. You have this very like filamentous kind of structure, and then it gets this single structure in the middle called I think it's called the rachis, and so then the little barbules kind of come off from the middle structure, which is the rachis, and then they get their own little barbules coming off of that. And so you can actually see the step by step transition of feathers. There's a lot of work done on the genetics of it. There's actually work shows that one of the genes for scales, I think, was duplicated a few times. And there's a paper on that that I should include in my video on aves because it will be a nice paper. So yes, there's a lot of work on that. Like I said, there was a feather morphotype that was predicted by embryological data, which was essentially a broad filamentous feather, and it was found in Bpiosaurus. That was something I mentioned earlier. So yeah, there's a whole lot of data out there on feather evolution. Just go to Google Scholar, type in feather evolution, find tons of Prum and Brush papers and lots of other stuff. So
you got it. Next, we have a question for standing from Dragnoct Silvis. And my guess is that the last question we might be able to get to, depending. Basically, he asks, since standings halo groups don't match with mainstream science, what techniques did he employ in order to determine the oh, I'm sorry, I pronounced that wrong. Since standings haplogroups don't match with mainstream science, what techniques did he employ in order to determine haplogroup M, as in Margaret, and N, as in Nancy, are lateral to L?
I'm sorry. I was answering that, but I was muted. That's okay. So I'm surprised he doesn't know that. I mean, using mitochondrial, it's pretty basic, using mitochondrial DNA and using direct measurements on many people groups worldwide, then lining them up will automatically reveal that map that I was showing on if you're looking at my opening presentation. Anybody can do it with hundreds of mitochondrial DNA sequences. I'm surprised he doesn't know. But he doesn't even understand the entropic degeneration of our genome, so I'm not surprised on that. But the evidence is certainly in regards to the three major nodes. The short lines really does indicate that these are three women that came from, say, a time where there was fewer generations, as well as the longer lines emitting from that. And we've actually, if you're looking at my presentation earlier, we've actually made testable predictions, because if we come from these three nodes, James, and the DNA differences are all a marker of time, we're looking here, what we're looking at here radiating from the three nodes would be post-flood times, which means the history of civilization. So we should be able to read the history of civilization from this tree, right? Roman Empire, Greeks, Persians. And we're actually looking at, Dr. Jensen is doing amazing work on the transatlantic slave trade. So at the end of the day, they can scoff at the data, but we've made the mitochondrial DNA predictions. We've made the predictions on the Khoisan peoples. Our model is testable and falsifiable and consistent with the empirical data. So until the evolutionists can actually, man up and make some testable predictions as well, given the data, I say it's premature to scoff at the work that's being done. Thanks.
So we have got a lot of questions that have come in, and we want to say thank you for all of your questions. If you're new here, hit that subscribe button so hopefully you can come back and see these debaters again, as I would love to have both of them. It's been a friendly debate. You guys just exercise so much self-control and maturity. Like it's honestly a pleasure every time. And so, as well as being so articulate and having hit the books, I am just so glad. So thank you guys so much. But for our debaters, their links are in the description, everybody. Totally encourage you, go like, subscribe their stuff. That way you can, even if you, maybe you're like, I'm not going to like their stuff. Well, subscribe and you can at least get it from their point of view. I mean, think about this. Isn't it practical? Even if you're like, I will never agree with Standing for Truth or Jackson Wheat, whoever it is, you might say, well, even if you're not going to agree, you might as well study your enemy in a way. And I mean enemy loosely, like a person you might disagree with. So at least understand where they're coming from. And we are glad you've come. Thank you for the debaters again, both Jackson and Standing for Truth, for coming tonight.
Thank you so much, James.
Thanks for having me. I appreciate it.
Oh, it's my pleasure. And those of you in the audience, by the way, let us know if there are new debate topics that you want to see. If you're like, oh, snap, I know somebody in the comments already dropped one. Somebody said, hey, we want to see an atheist and a theist debate on whether or not evolution should make us atheists. So that's a really interesting one. If you have any other ideas, please feel free to shoot me an email at moderndaydebate@gmail.com, or you can also put it in the comments. And last, we would just say that if you want to debate, that it describes how in the description box how you can come on. Basically, we have a calendar. You can look for an empty slot on Monday, Wednesday, Friday, Saturday, and we will try to get you on. So thank you very much, and we hope you have a great night. Keep sifting out the reasonable from the unreasonable. Have a good night.